Good morning, everyone. I'm delighted to welcome you to today's presentation. We will be exploring sterile filtration technology, with a focus on the scientific basis for 0.2 micron filters. Today's session will cover the fundamentals behind these filters, how they work, and their importance in ensuring sterile products. Let's dive in and discover the science that makes these filters so essential in our industry. Let's take a closer look at why sterile filtration is so important in pharmaceutical manufacturing. First and foremost, it ensures sterility assurance, meaning we can be confident that the products are free from harmful microorganisms. This directly protects patient safety, which is always our top priority. Sterile filtration also supports aseptic processing, helping us maintain a contamination-free environment during production. Additionally, it improves process efficiency and reliability by reducing the risk of contamination-related failures or delays. Of course, adhering to strict regulatory standards is essential, and effective sterilization methods like this help us stay compliant with those regulations. Now, to tie this into the overall theme, sterile filtration isn't just a technical step. It's a fundamental part of ensuring the safety, quality, and efficacy of the medicines we produce. Recent advances in membrane technology, design, automation, and integrity monitoring have further enhanced these processes, making them more efficient, reliable, and better at controlling contamination. These innovations are vital as we strive to meet the ever-increasing demands for quality and safety in our industry. When we talk about regulatory expectations, the first key point is the use of validated sterilizing-grade filters. These filters are essential because they provide a proven barrier against microbial contamination, ensuring the sterility of our products. It's not enough to just have filters. We need scientific justification for our filtration process. This means having solid data and rationale to support that our chosen method effectively removes contaminants. Integrity testing before and after filtration is also crucial. Performing these tests ensures the filters are functioning correctly and that no breaches have occurred during the process. This step helps us catch issues early and maintain confidence in our sterilization process. Controlling bioburden and contamination is another fundamental aspect. We need to understand and manage the microbial load in our raw materials and environment to prevent contamination from the start. Life cycle management and proper documentation are equally important. They help us track the filter's performance over time and ensure compliance with regulatory standards. Speaking of standards, there are several applicable regulatory guidelines we should be aware of. For instance, in the U.S., the FDA's 21 CFR Parts 210 and 211 set out manufacturing and control requirements. In Europe, the EU GMP Annex 1 specifically addresses the manufacture of sterile medicinal products. The USP 1229 provides guidance on sterilizing filtration, while PDA Technical Report Number 26 offers detailed technical insights. ISO 13408 outlines principles for aseptic processing, and the WHO GMP guidelines, along with PIC-S sterile manufacturing guidance, provide additional international standards. Connecting all these points, it's clear that compliance with these regulations isn't just about meeting requirements. It's about ensuring the highest quality and safety in our sterile products. Let's explore the scientific basis of 0.2 micron filtration. First, the principle of sterilizing filtration relies on size exclusion, meaning that bacteria and other microorganisms are physically blocked because they are larger than the filter pores. In addition to size exclusion, adsorption also plays a role, where contaminants stick to the membrane surface, helping ensure sterility. The tortuous pathways within the membrane create a complex maze that microbes find difficult to navigate, further enhancing filtration efficiency. Now, the scientific justification for using this 0.2 micron standard is based on the fact that most pharmaceutical bacterial contaminants are actually larger than 0.2 microns, making this pore size effective for removing them. When we consider challenge organisms, Brevendemonas diminuta stands out as the standard because of its small size and resilience. It's used to test and validate that our filtration process can reliably retain microbes. The main objective here is to demonstrate that our filtration system can consistently retain microbes even under the most challenging manufacturing conditions, what we call worst-case scenarios. This ensures that our products are safe and sterile, no matter the situation. Let's take a look at how sterile filtration technology has evolved over time. In the past, traditional filtration systems relied heavily on cellulose-based filters and depth filtration methods. While effective, these systems had some limitations, particularly in chemical compatibility, which could restrict their use with certain products. Now, moving into modern advances, we've seen significant innovations. Advanced polymer membrane technologies have emerged, offering better performance and broader chemical compatibility. These new membranes are designed for high flow rates and low protein binding, which is crucial for maintaining product integrity. Additionally, the industry has shifted towards single-use filtration assemblies. These are more convenient and help reduce contamination risks. Automated integrity testing systems also play a vital role, ensuring filters are functioning correctly without manual intervention. Another exciting development is the integration of smart filtration monitoring technologies. These systems provide real-time data and alerts, allowing for more precise control and assurance of sterility throughout the process. Overall, these advancements have had a profound impact on the industry by improving sterility assurance and operational efficiency, which are essential for high-quality biopharmaceutical manufacturing. This evolution reflects our ongoing commitment to safer, more reliable filtration solutions. Let's take a closer look at the selection criteria for membrane filters. When choosing a membrane, product compatibility is crucial, ensuring that the filter works seamlessly with your specific application and fluid type. Sterility requirements are also vital, especially in pharmaceutical and biotech settings, where maintaining a sterile environment is non-negotiable. Filtration efficiency is another key factor. It determines how effectively the membrane can remove particulates and microorganisms from your sample. Now, moving on to advanced membrane technologies, there are several common materials used in the industry. Polyethersulfone, or PES, is known for its excellent flow rates and chemical resistance. Polyvinylidene fluoride, or PVDF, offers high mechanical strength and is also highly resistant to chemicals. Nylon is another versatile option, valued for its durability and broad compatibility. And then there's PTFE, or polytetrafluoroethylene, which is prized for its inertness and high-temperature stability. Beyond the materials themselves, advanced features of these membranes really enhance their performance. For example, low protein binding properties are essential in many bioprocess applications because they help minimize sample loss. High chemical compatibility ensures that membranes can withstand aggressive cleaning agents and process conditions without degradation. Enhanced flow rates allow for faster processing times, improving overall efficiency. Reduced extractables and leachables are critical to maintaining sample integrity and meeting regulatory standards. Lastly, improved thermal stability means these membranes can perform reliably under varying temperature conditions, broadening their range of applications. All these factors, materials and features, connect directly to our goal of selecting the right membrane for specific needs, ensuring optimal performance, safety, and compliance in our processes. Let's take a closer look at the benefits of these advanced filtration technologies. First, you'll notice that increased throughput means we can process more product in less time, boosting overall efficiency. Reduced filtration time not only speeds up operations, but also minimizes downtime. Improved microbial retention is crucial for maintaining product safety and sterility, ensuring contaminants are effectively removed. Lower product loss helps us maximize yield, which is vital for cost management. Additionally, reducing filter fouling extends the lifespan of the filters and decreases maintenance needs. Now, the objective behind these innovations is clear. We want to enhance filtration efficiency without compromising sterility assurance. To achieve this, recent advances in filter design are key. These include asymmetric membrane structures, which provide optimized flow pathways and better retention. Multi-layer filtration systems offer improved clarity and purity by capturing a wider range of particles. High surface area cartridges increase filtration capacity, allowing for higher flow rates without clogging. Optimized flow distribution ensures uniform processing, reducing pressure drops and improving overall performance. Lastly, low differential pressure systems help maintain steady operation and reduce stress on the filters. All of these improvements are about making filtration more effective, faster, and more reliable, aligning perfectly with our goal of delivering safe, high-quality products efficiently. Let's take a closer look at some of the key advantages of using disposable sterile filtration technologies. First, you'll notice that setup and turnaround times are significantly faster. This means less downtime and more flexibility in your manufacturing schedule. Next, these systems reduce operational complexity. By simplifying the process, you can streamline operations and minimize the chances of errors. This also ties into enhanced contamination control, which is crucial in maintaining the integrity and safety of your products. One of the standout features of single-use systems is that they come with pre-sterilized assemblies. This eliminates the need for extensive cleaning and validation between uses, saving both time and resources. Additionally, these systems offer flexible manufacturing integration. They can easily adapt to different production processes, making them ideal for various applications. And because they are disposable, the risk of cross-contamination is lower, which is vital for products like biopharmaceuticals and vaccines. Now, speaking about applications, disposable sterile filtration technologies are especially valuable in biopharmaceutical manufacturing, where purity and safety are paramount. They're also heavily used in vaccine production, where rapid, sterile processing is essential. Lastly, they are perfect for small-batch sterile operations, enabling quick changes and maintaining high standards without the burden of extensive cleaning procedures. All these benefits collectively support the overall goal of improving efficiency, safety, and flexibility in sterile manufacturing environments. Let's now look at some of the latest innovations in integrity testing. One key area is advanced filter integrity verification. Here, we have methods like the bubble point test, which is a classic technique to determine if a filter is properly sealed by applying pressure until bubbles are observed. Then there's the diffusion test, which measures how gases pass through the filter to assess its integrity. And the pressure hold test, where we monitor the filter under a set pressure for a specific period to ensure it maintains its integrity without leaks. Moving on to technological advances, we're seeing systems become more automated. Automated integrity testing systems not only improve consistency, but also save time. Real-time monitoring and data logging are now standard, allowing us to track performance immediately and maintain comprehensive records. Integrated digital validation systems help streamline compliance, making validation processes more efficient. And with electronic audit trail integration, every test and result is documented securely, supporting regulatory inspections and quality assurance. Speaking of regulations, regulatory expectations have become more defined. For example, pre-use and post-use integrity testing, known as PUPSIT, is required per EU GMP Annex 1. This ensures filters are verified before and after use, reducing contamination risks. Additionally, there's an increasing emphasis on understanding the correlation between integrity testing and bacterial retention. Essentially, demonstrating that the integrity test results reliably predict the filter's ability to prevent bacterial passage is crucial for maintaining product safety and regulatory compliance. Overall, these innovations and regulatory expectations are pushing us toward more reliable, efficient, and compliant filtration processes, which are vital for ensuring product quality and patient safety. Let's take a closer look at smart filtration and automation. These days, digital and AI-enabled filtration systems are transforming how we manage process purity and efficiency. AI-assisted filtration monitoring allows us to keep a close eye on filtration performance continuously, helping us identify issues early on. Predictive maintenance systems are also a game changer, as they enable us to anticipate equipment failures before they happen, reducing downtime. Real-time process analytics give us instant insights into filtration parameters, ensuring optimal operation at all times. By leveraging IoT-enabled filtration equipment, we can connect devices seamlessly, creating a smarter and more responsive system. Automated deviation detection further enhances this setup by automatically flagging any anomalies, so we can act swiftly. Now, what are the benefits of implementing these advanced systems? We see improved process control, which means more consistent product quality. Reduced human intervention errors, thanks to automation, make processes safer and more reliable. Enhanced traceability and data integrity help us comply with regulations and provide clear records. And predictive risk management allows us to proactively address potential issues, minimizing risks before they impact operations. From a regulatory perspective, validation and secure electronic record management are crucial. They ensure that our filtration systems meet compliance standards and that records are reliable, tamper-proof, and easily auditable. These elements are essential for maintaining trust and transparency in our processes. Overall, integrating smart filtration and automation aligns perfectly with our goal of creating safer, more efficient, and compliant operations. Now, let's look at some key contamination control measures that are essential for maintaining bioburden and contamination control. First, we focus on controlled manufacturing environments. These are specially designed spaces that limit the presence of potential contaminants, helping us keep the process as sterile as possible. Next, low bioburden processing is crucial because it minimizes microbial presence right from the start, reducing the risk of contamination later on. Sanitized equipment and pipelines are also vital. They must be thoroughly cleaned and disinfected regularly to prevent any microbial buildup. Environmental monitoring programs play a key role here too. By continuously checking air, surfaces, and water, we can detect and address contamination issues early. And finally, timely filtration after preparation ensures that any remaining particles or microbes are removed before the final product moves forward. From a regulatory perspective, integrating filtration into our contamination control strategy, or CCS, is emphasized. This integration helps us meet strict industry standards and ensures our processes are robust. The main objective of all these measures is to reduce microbial load significantly and to maintain aseptic process integrity. In essence, our goal is to create a safe, sterile environment where contamination risks are minimized at every step, safeguarding the quality and safety of the final product. Let's now move on to the topic of validation and lifecycle management. First, we'll look at validation activities. These are essential to ensure our filtration systems perform consistently and effectively throughout their use. For example, bacterial retention studies help us verify that the filters can reliably remove bacteria, which is critical for product safety. Product compatibility assessments are also key. They make sure that the filter materials won't negatively interact with our product, maintaining its integrity. Extractables and leachables studies are conducted to identify any potential contaminants that could migrate from the filter into the product, ensuring safety and compliance. Another important activity is the filter integrity test correlation. This involves confirming that the integrity tests we perform are truly indicative of the filter's actual performance. Lastly, process simulation studies mimic real-world conditions to validate the entire filtration process under typical operating scenarios. Now, moving on to lifecycle management. This is about maintaining our validated state over time, even as conditions change. Change control processes are vital here. They help us manage any modifications to the system or process without compromising validation. Periodic review and revalidation are also necessary. They ensure that our systems continue to meet their intended performance. Supplier qualification is another crucial aspect. Working with reliable suppliers ensures the quality of the filters we use. Finally, trending of filtration deviations allows us to identify patterns or issues early, so we can address them proactively. The main objective of all these activities is to maintain a validated state throughout the product lifecycle, guaranteeing ongoing safety, quality, and compliance. This continuous approach is fundamental to our overall quality management system. Let's take a closer look at some common regulatory observations that we often encounter. First, there are frequently observed deficiencies, such as inadequate filter validation. This is critical because if filters aren't properly validated, it can compromise the entire sterilization process. Another common issue is missing integrity testing records, which makes it difficult to verify that filters are functioning correctly over time. We also see poor bioburden control, which means the levels of microbial contamination aren't being effectively managed. This can pose serious risks to product safety. Incomplete extractables and leachables studies are another concern. Without comprehensive data, we can't be sure that materials used won't release harmful substances into the product. Weak change control management is also frequently reported. Proper change control ensures that any modifications to processes or materials are thoroughly evaluated and documented, reducing unintended impacts. And finally, insufficient documentation and traceability can make it challenging to track the history of a product or process, which is vital during investigations or audits. Now, what are the potential consequences of these deficiencies? They can lead to GMP observations, which may result in regulatory inspections and potential citations. More seriously, they can trigger FDA warning letters, which can damage a company's reputation and lead to increased scrutiny. In some cases, these issues might even cause product recalls, affecting supply and trust. Most importantly, these deficiencies can compromise sterility assurance, risking patient safety and the efficacy of the product. So, understanding and addressing these common issues is essential for maintaining compliance and ensuring the quality of our products throughout the entire life cycle. Let's now focus on strengthening our sterile filtration systems through best practices. First, it's crucial to use validated sterilizing-grade membrane filters to ensure the highest level of sterility. Routine integrity testing is another key step. It helps us confirm that filters are functioning properly before and after use. Applying risk-based contamination control strategies allows us to prioritize efforts where the most critical risks exist, making our processes more efficient. Conducting periodic filter revalidation ensures that filters continue to meet performance standards over time, especially after changes or maintenance. Integrating automated monitoring systems can provide real-time data, enabling quicker responses to potential issues. Training personnel on aseptic filtration practices is fundamental. Well-trained staff are our first line of defense against contamination. Maintaining complete documentation and audit trails not only supports compliance, but also helps us track performance and identify areas for improvement. Additionally, staying updated on emerging filtration technologies and regulations ensures our practices remain current and effective. Moving to continuous improvement, it's vital that we trend filtration failures and deviations. By analyzing these patterns, we can identify underlying causes and prevent future issues. Reviewing process capability and filter performance regularly helps us gauge how well our systems are performing against our quality standards. When recurring issues are identified, implementing corrective and preventive actions, or CAPA, is essential to address root causes and prevent reoccurrence. Together, these practices form a comprehensive approach to maintaining and enhancing the integrity of our sterile filtration systems, which ultimately supports the overall quality and safety of our products. Let's wrap up with some key takeaways. First, 0.2 micron membrane filters continue to be the gold standard for sterile filtration, both scientifically and regulatorily. They've proven their effectiveness over time. Advances in membrane materials, automation, and smart monitoring have really boosted filtration efficiency and reliability. These innovations help us ensure consistent sterility and save time. Robust validation and lifecycle management are also crucial. They're essential for maintaining sterility assurance throughout the product's lifecycle, reducing risks and ensuring quality. Integrating filtration into broader contamination control strategies helps strengthen GMP compliance. It's about building a comprehensive approach to quality and safety. Finally, emerging technologies are continually pushing the boundaries, improving operational efficiency, enhancing product quality, and ultimately protecting patient safety. All these points highlight how ongoing innovation and rigorous practices are key to maintaining high standards in sterile filtration. For further support, please feel free to contact us. We're here to help you with any questions or additional assistance you might need after this presentation. Remember, your success is our priority. Thank you so much for your time and attention today. We appreciate your engagement and look forward to supporting you in the future.